Welcome to Healthwise Show. Today, we're diving into the fascinating world of liquid chromatography. Liquid chromatography is an analytical technique used to separate ions or molecules dissolved in a solvent. It's a critical process used in various scientific fields from pharmaceuticals to environmental science. Stay tuned to learn about the different types of liquid chromatography, how they work, and their amazing applications. First up, let's explore ion exchange chromatography. Picture this. You have a tube packed with a special resin. Think of this resin like a bunch of tiny magnets. As our mixture of molecules travels through the tube, some are attracted to the resin due to electrostatic forces. Now here's where the exchange part comes in. We can introduce a special solution that changes the charge of either the resin or the molecules. This change disrupts the attraction, causing the molecules to break free and flow out of the tube. Depending on what we're trying to separate, we can use different types of resins. Anionic resins attract positive ions, while cationic resins attract negative ions. Ion exchange chromatography is like a powerful sorting machine for molecules. It's used in applications from purifying water to analyzing complex biological samples. Now let's dive into Gel filtration chromatography, also known as Gel permeation or molecular exclusion chromatography. This type of chromatography is all about size. Imagine a big playground slide. Kids of all sizes line up at the top, eager to slide down. As they go down the slide, the smaller kids get caught up in the twists and turns, while the larger kids slide right through. Gel filtration chromatography works similarly. We use a porous gel packed into a column. Smaller molecules enter the tiny tunnels and pathways, while larger molecules travel a shorter distance. As a result, the larger molecules come out of the column first, followed by the smaller molecules. This technique is incredibly useful for separating molecules of different sizes, such as proteins, DNA fragments, and polymers. So there you have it, Gel filtration chromatography, a simple yet elegant technique that reminds us that sometimes size does matter. Chapter 3. Affinity chromatography, finding the perfect match. This method is like a matchmaking service for molecules. It's all about using highly specific interactions to capture and isolate our target molecules. In affinity chromatography, we create a stationary phase that contains a molecule called a ligand, designed to attract our target molecule. When we pass a mixture of molecules through our column, only the target molecule binds to the ligand, like a perfect match. All the other molecules simply pass through the column. Once we've captured our target molecule, we can release it from the ligand by changing the conditions in the column, such as the pH or salt concentration. Affinity chromatography is used in various applications, like purifying antibodies for medical treatments, isolating enzymes for industrial processes, and studying protein interactions in cells. Chapter 4. Partition chromatography, a balancing act. Let's explore the world of partition chromatography. It's like a delicate dance between two phases, the mobile phase and the stationary phase. In this technique, the stationary phase is a thin liquid film coated on a solid support, while the mobile phase is a liquid that flows over the stationary phase. As our mixture of molecules travels through the column, they constantly interact with both phases. Molecules more soluble in the stationary phase will spend more time clinging to the solid support. On the other hand, molecules more soluble in the mobile phase will be carried along faster. This difference in affinity causes the molecules to separate as they move through the column. Partition chromatography is a versatile technique used in various applications, from separating and identifying drugs in forensic science to analyzing food components in the food industry. Chapter 5. Adsorption chromatography, sticking to the surface. Get ready to explore adsorption chromatography, where molecules play a game of stick or slip with a solid surface. In this technique, we use a solid stationary phase packed inside a column. When we introduce our mixture of molecules into the column, some molecules are attracted to the surface of the solid phase and stick to it, like kids clinging to a jungle gym. As the mobile phase passes over the stationary phase, it tries to carry the molecules along with it. However, some molecules are more strongly attracted to the surface and resist being carried away. This difference in attraction creates a separation between the molecules. Those that are strongly adsorbed to the stationary phase move slowly, while those that are weakly adsorbed move faster. Adsorption chromatography is a powerful tool for separating and purifying a wide range of compounds, from small molecules like gases and pigments to larger molecules like proteins and nucleic acids. Thanks for watching. We hope you now have a better understanding of liquid chromatography and its various types, from separating ions based on charge to sorting molecules by size and utilizing highly specific interactions. Each type of chromatography offers a unique way to analyze and purify complex mixtures. Don't forget to like, subscribe, and hit the bell icon for more informative videos. Until next time, stay curious.